The recording on? Yeah, the recording is on. All right, good morning, students. So today we are going to look at another kind of molecular motor. So we have so far looked at polymerization motors like actin, tubulin. We have looked at motors that generate translocation forces such as myosins, dyneins, and kinesins. We have studied rotary motors such as bacterial flagella, and we have also looked at ATP synthase. And we have seen that how these two, ATP synthase and bacterial flagella, are driven by proton gradients. They are driven by proton gradients to cause the torque that induces the rotation of the flagella, or in the case of ATP synthase, that causes the rotation of or the movement of the subunits that causes the final, what should I say, change of configuration of the head subunits that finally leads to production of ATP. Today we are going to look at another class of motors called helicases. And I'm sure you all know something about helicases. So before we begin, let's hear what you all know about helicase. So for that, I will start with Ratnesh because he's the youngest member in the team. Ratnesh, what do you know about helicase? When you hear helicase, what is it that you think of?
Ma'am, helicases are like it is in we see that in while DNA replicates yes no no yes, in DNA replication what it does is it unwinds this DNA.
Perfect, perfect. Ratnesh has said that he's heard of helicases when he was studying DNA replication, where he came to know that in DNA replication, helicases are responsible for unwinding DNA. Then let's go to Kavan. Kavan, what do you know about helicases? Is there something more that you would like to add to this?
No, that's what I know also.
All right. Anybody else? Farman, Sarika, would you like to add something more? Do you know something else? Yes, Sarika.
Yes, ma'am. One thing what Ratnesh has missed is it uses ATP hydrolysis activity to separate the two strands of nucleic acid, and it is not always DNA strands. It can be RNA strands also.
Very good, very good. So Sarika has pointed out that it would require, for this kind of an activity, it would require some source of energy. So that could be ATP, and that could be ATP. It's one of the sources. It could be any other nucleotides too. We would see that. And she has pointed out. What's the second thing you said, Sarika? Sorry, I forgot.
Ma'am, Ratnesh told that it is
RNA.
DNA
Right, right,
strands.
right. So what Sarika says that it's not just in DNA replication. That means it's not just DNA, double-stranded DNA, which can be unbound or unzipped by the helicases, but it could also be RNA, which has formed these double-stranded structures, local double-stranded structures, double-stranded RNA, whatever is the case. Thank you, Sarika. Anything else you would like to add, Sarika?
And ma'am, it's a motor protein.
Yes. Is this kind of touching upon your PhD subject, your PhD topic?
Uh, not not properly, but the protein I will be working on is belong to one of the helicase.
Okay, okay, perfect. Thank you. Farman, would you like to add something to this?
Yes, ma'am. Actually, it has six subunits, which and it is also a triple A ATPase family protein.
Your voice is not clear, Farman, for some reason. I can't hear you clearly.
Ma'am, it has six subunits and it has triple A ATPase activity. You know,
It has 6 units and
and it is triple A ATPase protein.
okay,
Belongs
okay,
to triple A ATPase.
okay, okay, all right, thank you. Anything else you'd like to add?
Mm,
No?
no, ma'am. No.
All right, thank you, thank you. So we have collected a lot of information, and this kind of sums up everything that I was about to say in this lecture. So helicases are a group of proteins, vast group of proteins. It's not one or two, and in fact, in our bodies, there is not one or two, there are multiple types of helicases, as pointed out by Sarika and pointed out by Ratnesh. They are required for multitude of functions. So let's quickly go to, I mean, this is kind of a repetition, but I thought it's important to just look through it so that we recapitulate what is DNA and what is RNA, because these helicases work on DNA and RNA. So DNA and RNA, they have something called. So if you look at the backbone structure, you would see that there is a ribose or a deoxyribose sugar, and attached to that is a nitrogenous base. So these four structures here are the structures of nitrogenous bases that are present in DNA. Now in RNA, one of this would be slightly altered. Can somebody point out which of these nucleotide bases would be slightly altered and what would be the alteration?
Ma'am, it will be thymine, and I remember like there is this CS3 will not be there.
Okay, and what would that be called?
Uracil.
Okay, thank you. So you have these four nitrogenous bases, these purines, these pyrimidines, and in RNA, instead of thymine, you would have a uracil. So these are the four nitrogenous bases. And why are we concentrating on the nitrogenous base? Okay, by the way, this is how the nitrogenous bases would be linked to the sugar moiety. So the sugar moiety could be deoxyribose, as is shown here, or it could be just ribose, in which case this hydrogen, this H would be OH. And along with this ribose or deoxyribose, you have this phosphate, which forms which links with the next nucleotide. So in this way, what it forms are these kind of polymerized chains, and each of these polymerized chains is actually protruding one of these nitrogenous bases towards the inside of a double helix. So this is the structure of a DNA double helix. And what you see here is that there is very specific base pairings that can occur in a DNA double helix. And for the most stable structure of a bDNA, you would see that an adenine would have two hydrogen bondings formed with a thymine, and a guanine would form three hydrogen bonds with a cytosine. In this manner, you would have the formation of a double-stranded DNA. You would also have double-stranded RNA, again by the same principle of forming hydrogen bonds between the base pairs. So what is holding the double-stranded structures is basically the formation of the hydrogen bonding, and the next thing is the stacking interaction between these ring-like structures, because they are planar ring-like structures, so there would be stacking interactions between these bases, the base on the top, and so if you take this, the base on the top and the base of the bottom, stacking interactions as well as hydrogen bonding interactions. So if you need to form a single stranded DNA from this double stranded DNA, if you need to take these two DNA strands apart, you need to provide energy, right? That's the first thing. You need to provide energy because this is a very stabilized structure. You need to provide energy to break these bonds, to break these interactions. Now, in laboratory, when we do polymerase chain reaction or PCR, you all know about PCR, right? Is there anybody who does not know PCR? Please raise your hand if you do not know. No? Okay. So I guess all of you know about PCR. So what happens in PCR? You provide a starting material, which is usually a double-stranded DNA, and you provide primers that would be complementary to the region that you want to amplify. And in order to have these primers anneal to your DNA, you need to separate out the two strands of DNA. So what do you do to separate out the two strands of DNA in PCR?
Ma'am, we do like we heat this DNA.
Right. You heat the DNA. That means you're providing it enough energy to overcome these hydrogen bonds, to overcome these base stacking interactions. But inside a cell, when, say, for example, very simple analogy of DNA replication, when DNA replication takes place, then you do not expect there to be a change of temperature. Everything is happening at the temperature at which the cells are residing, the cells are growing, which is nearly roughly about 37 degrees. So at that temperature, though a temperature of 37 degrees is not enough to cause separation of the two strands. So how is the separation of the two strands achieved inside the cell? So there must be a mechanism that separates out the two strands. And the mechanism that is used is an energy driven mechanism by utilizing proteins called helicases. So this is a cartoonic representation of DNA replication. It's going to tell you exactly the same thing that I have described so far. What you see here is a DNA double helix, which has A base pairing with T, G base pairing with C. And when this DNA double helical strand has to be replicated, the first step is to ensure that the two strands separate out. And this is done by a protein called helicases. So exactly what I have described is being shown to you. Now, here this is important. What you saw in before is that the two strands have a polarity, a 5' end and a 3'. Now, do you understand what this 5' and a 3' end actually means?
Yes, ma'am. At 5 prime end, there is a phosphate group, and the 3 prime end is OH group is ends with OH. Yeah.
Right. So when there is polymerization at one of the phosphate group at the end would be free, would not be bound to another nucleotide, whereas at the other end, a hydroxyl group would be free and would not be bound to the next phosphate. So that causes the presence of a 5' end and a 3' end. So in a double helical DNA, in a double stranded DNA, you would have one strand running from 5' to 3' and the complementary strand running from 3' to, this side would be 3', this side 5'. So 5' to 3', 5' to 3'. All right. So this is your helicase that is, you found that it was moving on the DNA, kind of separate DNA. We're not going to the next. So this idea that you need something to separate out the two strands of DNA was very evident because you need a way to replicate DNA. The polymerases, DNA polymerases, RNA polymerases were identified, but it was not quite known what is causing this separation of the DNA. So people were looking for components. So they took cells, they were fractionating the cells, obtaining different different fractions of protein, different sizes of protein or protein complexes, and trying to see which of these protein complexes or which of these fractions can cause separation of either double-stranded DNA or a DNA RNA complex hybrid. And this paper is actually the beginning of research on helicases. So this is around 1970 and it shows 76 here. So what they did was they were looking at, as I mentioned, they had no idea what is this protein, or it's a protein complex, or it's a single protein, or multiple proteins coming together. So they took E. coli cells, bacterial cells, E. coli cells. They burst open those E. coli cells, and whatever proteins you have, you can now try to separate them out according to their size or some property. It could be size, it could be hydrophobicity or some kind of property. And they obtain these different fractions. And one of the fractions showed them some ATPase activity, an ATPase activity, and a strand separation activity. So what you see in this figure is a very simple thing. What they did was they took a DNA RNA hybrid, one strand of DNA, one strand of RNA, which is complementary to the DNA strand, so it can form base pairs and it can form a DNA RNA hybrid. Such hybrids are formed during replication. If you remember, I stopped at the point where the primase had come and bound to the single strand DNA. The primase is known to produce a primer, and in cells, the DNA replication process requires a primer, which is an RNA molecule. So you have a DNA RNA hybrid. You remember this? You all
Yes,
remember?
Yes, ma'am.
Yes? Okay, so here similarly they designed it. This is a designed piece of DNA and a complementary piece of RNA, and this complementary piece of RNA forms double strands with the DNA. Now, they specifically radio labeled the RNA. This H3 is tritiated hydrogen, and this would help them identify the RNA and where it is present. So you can understand 1976 was a time when they did not have a lot of different tools, but they had a means to identify whether the RNA is not bound to DNA, or that is whether the RNA is free or the RNA is bound to a DNA RNA complex. For details, you could have a look at this paper. So what is essentially shown here is that they take this DNA RNA hybrid and they add this fraction, the fraction of the protein where they believe it has an activity to cause separation of the DNA and RNA hybrids. And they see that as the time proceeds, the RNA gets detached from the DNA, and so you do not see a DNA RNA hybrid. You see a decrease in the concentration of DNA RNA hybrid. And because the single stranded RNA can be removed, you see a decrease in the tritiated RNA levels with time. I repeat this. So what they did is they took the fraction which they believe had this activity of separating a DNA RNA hybrid. They have used a radioactively labeled RNA strand, and now the moment they add that fraction, which they believe has kind of a helicase activity, they see that the levels of this DNA RNA hybrid goes down. And that is what they have plotted here. They see that the levels of this DNA RNA hybrid is going down. What they also noticed is that this activity is dependent upon magnesium ATP. So if you leave out ATP, you see that this activity does not take place. On the other hand, if you add ATP, but if you don't add magnesium, and you add something which is a chelator of magnesium, that is, even if there is a trace amount of magnesium, it can chelate out, or it can remove it out of the solution, keep it in a coordinated, what should I say, form a coordination complex with magnesium, and therefore magnesium is not available for the reaction. The stuff is called EDTA. And if you use this, again, there is no reaction. So this helicase activity, what they found was it is dependent on ATP, some energy source, and it is dependent on magnesium, presence of this divalent magnesium ion. So this was the start and identification of helicases. Since then, helicases, as I've said, they have been studied a lot, and what has been found is that there is really a huge number of helicases. They can take part in a number of processes. Their structures can be quite varied. They can be monomers, dimers, heterodimers, homodimers. They can form hexamers. So there is, and just as I think Farman had mentioned, AAA ATPase kind of proteins, there's a multitude of them. What connects them are two things. One is that they all have a, I would not call it ATPase because it may not be just ATP. It could be um, any triphosphate nucleotide. It could be ATP, GTP, even TTP. And I'm going to give an example at the end where TTP is used to drive helicases. So it could be any of these um, uh, triphosphate nucleotides, and that would be the source for these helicases to function. And then the second aspect is that there would be an associated conformational change that would be driving this reaction or causing this motor to function. So one is the energy of hydrolysis of the terminal phosphate from any of the four ATP, GTP, TTP, CTP, be any of them. The second is that there would be an associated conformational change. So again, we come back to the same thing that we had learned in the very first class, how motor proteins function. You need a source of energy, and that would be driving an associated conformational change. Okay, so helicases, as we have learned, they would not just be working on DNA, but they could also be working on RNA, right? In this example itself, you've seen that it works on DNA, RNA duplexes. It could also be RNA duplexes, DNA duplexes, etc. So helicases can function on both DNA or RNA. And helicases can use any of the NTPs for the hydrolysis, the energy of hydrolysis that is obtained by removing the terminal phosphate from any of the NTPs to drive the unzipping of DNA. And they take part in a number of processes. Now, we have learned about DNA replication. I've already mentioned a few here, like DNA repair, recombination, transcription. Do any of you know anything else that we could add to this? Sarika, any other function that I have missed out? So whatever I've listed here, everything pertains to DNA. When there is replication, you need to unwind the two strands. If there is a mismatch and that needs to be repaired, again you need to unwind the two strands. Transcription, you need to unwind the two strands. You need some kind of a helicase activity there. Anything else, recombination, you would need, if there is a formation of a Halliday junction, you must have read about that. Then you can have a helicase that can function just at the junction. Anything else that you can think of, Sarika, that I have missed?
Splicing, what I have studied about my protein, it is involved in all processes of RNA metabolism, which includes splicing, unwinding any link related to RNA.
Okay, okay. So what you're saying is that when the heteronuclear RNA is formed that has all the introns and exons intact, and when splicing reaction has to take place, how is that, where is that required in the splicing reaction? If you could just give 2 lines about it.
In the process of catalyzing this reaction for the separation of the for the breakage of the introns among the exons, so for to denature those nucleotides which are a part of introns, it is required in that step.
So would it be required because the introns and exons form inherently secondary structures? They would form those loop-like base pairings, or is it required to actually bring those I don't know how it's required for the looping and opening of the loops so that the two exons are brought close by. That's what you mean?
Yes, ma'am.
Okay.
Yes, ma'am.
Okay, thank you, Sarika. So, as you've seen, there is a multitude of processes, and as you can very well understand, not all helicases would do all of these functions. if the same helicase were to do all of these functions, it would be very difficult to regulate. So one way by which nature regulates these activities is by producing a number of different helicases that can individually do all of these functions. Indeed, if you look at the human genome, you will see, I'm not quite sure, but maybe 70 to 90 such helicases that are present in human genome, which is quite a number considering that they're all actually helicases. So coming to the next point, helicase structure. So if you look at the structure, the number of subunits that might be present in helicases, that's also very diverse. Now helicases, the first example of helicases or the first identification of helicases was done in E. coli, which is a bacterium. But I have said, and Sarika has also mentioned, that she works on helicases that are present in eukaryotes. So from prokaryotes to eukaryotes, all of these organisms have helicases, and the helicases could have all of these combination of structures. So you could have a single monomeric helicase, just one monomeric unit, or you could have formation of dimeric helicases, two dimers. It could be a homodimer, it could be heterodimer. You could have formation of higher oligomeric structures. So this is just a representation. Do not think that it's one helicase after the other. It could have a higher oligomeric structure, which might have a functional consequence. So it might be a functional oligomer. That is, one oligomer help one unit helping the other, and then all of them combining together to cause unwinding of DNA, RNA, whatever is the case. Or it could be this kind of a hexameric structure. And when I say hexameric structure, we have already studied this kind of hexameric structure in something called AAA ATPases. Now, where did we study AAA ATPases? In what context? Come on, students. We studied AAA ATPases. In what context?
In dining, I think we studied this.
Huh?
Maybe dining we studied this.
In
Am I
dining.
audible?
In
Yes,
dining. That's
ma'am.
what you said. Dining.
Yes, ma'am.
And what was the AAA ATPase structure doing in dining? To all of you, what was the AAA structure doing in dining? What kind of catalytic function did it have? See, the name itself says it. AAA ATPase. क्या function आएगा उसमें? ATPase, ATP hydrolysis,
Yes, ma'am.
right? Okay, all right. So please study this dining thing because dining would be part of your major exam. Dining आएगा आपके major exam में. Okay, so in AAA ATPases, you have a hexameric orientation, a ring-like orientation of the subunits. So the orientation looks almost like a donut, except that instead of having a continuous ring, you can think of individual 6 subunits that are present in a ring-shaped fashion with a central hole, and that creates your AAA ATPase structure. And the ATPase function is present at the interface between two subunits. It's present at the interface between two subunits, and in dining we had seen that not all these AAA ATPase subdomains have an ATPase activity, whereas in this case we would see that each of these subunits have an ATPase activity associated with it. So for hexameric helicases, you can have 6 individual subunits arranged in the form of in the shape of a donut, and each of these subunits would have an Not necessarily an ATP. As I said, it could be any nucleotide triphosphate hydrolysis activity. The example that we will see today will have a TTP hydrolysis activity. So it could be any of these NTP hydrolysis activity, and usually the hydrolysis activity is at the interface between two supplements. All right. Helicases can work in either 5' to 3' direction or 3' to 5' direction. And this is very important because you have, if you just think of a single stranded DNA, it's easy to understand that it moves from 5' to 3' or 3' to 5'. It can understand which way it has to go. But now think of a double stranded DNA. And now if a molecule is looking at the double stranded DNA, तो फिर इस साइड से देखेंगे तो 5' to 3' यहाँ से है, इस साइड से देखेंगे तो 5' to 3' यहाँ से है. How does it identify which strand it has to bind and on which strand it has to make its activity going? So there is some amount of you understand the point, right? What I'm trying to mention, that you have a single strand and you now have to identify which is the 5' and the 3' direction. It's easy to identify. You have two strands. आप इस तरफ से देखेंगे यहाँ से 5' 3' वहाँ से 5', so कौन से strand पे आपको bind करना है? So these are kind of questions that a helicase has to encounter when it looks at a double-stranded DNA or double-stranded RNA structures. So we will look at it in a little more detail tomorrow. But for today, let's just understand that it could go in either of the direction. It depends on the kind of helicase, whether it goes from 5' to 3' or 3' to 5'. It's kind of analogous to kinesins, that in general we think that kinesins always move from inside the cell to its periphery. Therefore, it's moving from the negative end to the positive end on the microtubules. But we do know that there are kinesins that could walk in the reverse direction too. All right. Okay, so here are two theories on how helicases might function. So, what are the two scenarios by which helicases could cause unwinding of, let's say, DNA? So, we'll take a very simple example of DNA as being the structure that has to be unwound. So, a double-stranded DNA is present. Now, how would helicase start to unwind this DNA? The first idea that was put forth was that of passive unwinding. So what's passive unwinding? So if there is a double-stranded DNA, there is a chance that ever so often one or two of these base pairings can be disturbed because of thermal fluctuation. And this can create a very local disturbance on a double-stranded DNA because of thermal fluctuations. There is a chance that one or two base pairs could be disturbed. And the helicase might identify those local disturbances and start unwinding the DNA from that point. So this is one hypothesis. It's called the passive unwinding mechanism. But what's the other hypothesis? The other hypothesis is called the active unwinding hypothesis or active unwinding mechanism, where you consider that there is already a kind of a fork or a point where DNA has melted. You know what melting means? Melting means the unwinding of a part or completely the two strands of DNA, of double-stranded DNA, could also be double-stranded RNA. So if there is a partial unwinding that creates a fork-like condition, then the helicases could identify that fork and start unwinding this DNA even further. And it is possible that both of these mechanisms could coexist together, that there is a small thermal fluctuation that is identified and that causes formation of this small fork. So initially it is a passive mechanism by which the helicase is recruited, and then once this fork is produced, this fork can be actively what should I say? The helicases can start working on this fork and using the energy of hydrolysis of NTP start unwinding the DNA. Okay, so here are certain mechanisms of strand separation, and it's more or less what I have said so far. It could be, as I said, it could be a passive mechanism or an active mechanism. And then how does it go about it? So in one of these cases, you can see that the helicase is bound to one of these strands at the fork, and then it starts to unwind or unzip this double-stranded helix. As it does this, it translocates the single-stranded DNA through it, and then as it translocates, it kind of unzips the entire DNA, and you have formation of longer stretches of these single-stranded DNA. For this kind of a function, it needs to identify one of the strands and bind to it, depending on what is its affinity. So whether it is a 5 prime to 3 prime moving helicase or a 3 prime to 5 prime moving helicase, it could choose either of the strands, and it could start actively unwinding this DNA. How does it do it? It simply it keeps pulling the DNA, single-stranded DNA through it, translocates the DNA, and it unwinds the DNA. Another idea is that binding of the helicase to the DNA could cause local disturbances in DNA structure. So this is here, you see the DNA is bound here, and this has caused this local unpairing of the DNA structure. And so as the DNA is translocated through it, it creates more, it sort of moves this helicase to fresh places on the DNA, thereby creating local disturbances, local unwinding, and this carries on. So these are two mechanisms. There are four other mechanisms that are proposed, but this mechanism is proposed for helicases which can function as monomers, dimers, homodimers, or heterodimers. Helicases that can function as hexamers could behave in a slightly different fashion, and we'll come to that. But so far, do you have any questions? Any questions so far, or is it okay?
Ma'am, I have one
It's okay.
question. You mentioned
Yes,
you
go ahead.
mentioned that
Rakesh, you
there
can
are
start.
17 to 19 helicases. So, is this in a total genome or in each chromosome?
That's a good question. This I'm not quite aware of. I I suspect it should be scattered. You want to know whether the genes are present on just one chromosome or scattered throughout the human genome. I suspect they should be scattered throughout the human genome, but I'm not sure. I will have to recheck and get back to this.
Yes, ma'am. Okay, ma'am. Like I think like each and every chromosome must have like one helicase at least.
No, no, no. What's your question? I thought you're asking about the genes
Yes,
that
ma'am.
code
Yes.
for the helicases, whether they are present at one region in the chromosome or scattered throughout. I suspect they're scattered throughout, but I do not know the exact answer. I will cross-check, verify, and get back with the answer. Uh, but the idea that each of the chromosomes should have associated one specific helicase, I do not think that is the point here. Because you can have helicases with different activities. You could have repair activities, recombination activities. You could have activities which are not at all associated with DNA but associated with RNA.
Yes, ma'am.
Okay. Sarika?
Yes, ma'am. I was saying it's okay.
Oh, it's okay. All right. All right. Let me go ahead. So, if we now look at how these hexameric molecules or hexameric helicases behave, that's more interesting because you can think of a lot of possibilities. So if you try to see the cartoonic representation of helicases, specifically the ones that I have shown before and the one I would show now, you would see that there is there is a hexameric structure, and you can have the double-stranded helicase, the double-stranded DNA come into the groove from one end, and from the other end you see two single-stranded DNA structures coming out. So it's as if the whole helicase is pulling the DNA into it and then spewing out these single-stranded structures. So the helicase could be loaded onto this double-stranded DNA in this kind of a hexameric thing. The spewing out, or it could be loaded on these single-stranded structures. It could be either. It could be loaded on the single-stranded structures. It could be loaded on double-stranded structures. It could be pulling the DNA into it and releasing the single-stranded DNA structure. It could itself have a torsional activity, so it's spinning, and as it spins, it unwinds the DNA. It could cause the DNA single strands to come out from the central hole, or the DNA single strands might be simply splayed apart by some structure which has been described here in this arrow. It's as if the double-stranded DNA structure is getting into this hexameric ring, but there is something that's sort of pushing the two strands apart, like clawing the two strands apart. So there are different ways by which people have tried to envision how these hexameric structures could be working on double-stranded DNA, or it could also be double-stranded RNA. These are just cartoonic representations of all the different ways by which it could work. It could be loaded on double-stranded DNA, loaded on single-stranded DNA. It could cause a torsion, and therefore as it rotates, it's sort of unwinding the DNA, or it could be pulling the single-stranded DNA through it, or it could be pulling the double-stranded DNA, and there is something in between that could be splaying apart the two single-stranded DNA. So there's all kinds of possibilities, all kinds of theories, because there are multiple of these AAA ATPase like helicases that are present. So one such interesting helicases that I'm just going to highlight here is the T7 helicase. Now, T7 is a bacteriophage. I hope you've all read something about bacteriophages. So bacteriophages would have their own DNA, and they would also have some associated proteins that would help them replicate when they are infecting E. coli. So this T7 has its own ligase, has its own helicase. So this is a T7 helicase. And T7 helicase does a translocation from the 5' end to the 3' end on single-stranded DNA. And it utilizes the energy of hydrolysis of, sorry, this is not D, this has to be double strand, this has to be TTP. So utilizes the energy of hydrolysis of thymine triphosphate. And with this, it causes the separation of the two strands of DNA. Now, T7 forms a ring-like hexameric structure. And we would see how this ring-like hexameric structure works in this video. So, play this video. I think with this, I'll stop the class for today. So here is your fork, and the T7 hexamer has loaded onto the fork. And this is how, if you look at the structure or the crystal structure of this hexamer, this is how it would look. So you have all of these 6 subunits. Each of them have the capacity to bind to this nucleotide triphosphate and to hydrolyze the nucleotide triphosphate. But at a time, not all of these subunits would be bound to the same, would be in the same conformation. For example, these two would be bound to ATP. These two would have hydrolyzed, not ATP, sorry, TTP. These two would have hydrolyzed TTP, so they would be bound to TDP and inorganic phosphate. Whereas some other subunits may have released the hydrolyzed DNTP. So here they are mentioning it with ADP, ATP, but if you're thinking of it in terms of T7, think of it that you need TTP. So what is this energy of hydrolysis of the nucleotides doing? It is causing local changes in conformation of these subunits. And therefore, it is causing changes in the position of the loops that are in contact with DNA. It's almost as if the entire structure is dancing, and these changes in the position and orientation of the loops seems to be changing the way the DNA is bound to this hexameric structure. And it is believed that this is what is pulling or translocating the DNA through this hexamer. So again, you have this hexameric complex that is loaded onto the DNA. What is crucial here is to understand that all these six subunits can have the will have an associated hydrolysis activity. You see, it is the hydrolysis activity is close to the interface between two entities. But not all subunits would be bound by ATP at the same time or bound by not ATP like NTP or NDP plus PI or MT at the same time. So you can think of it as having two subunits. This entire idea comes from crystal structures. So what they found is that you can have different conformations of these individual subunits because the individual subunits might be bound to different nucleotide states. So they could be bound to NTP, they could be bound to NDP plus PI, or they could be empty. In the process, they would give rise to different conformations of the loops that are associated with DNA. So at a time, if you look at these conformational changes, if you have a top view on this ring, you would see that the ring is almost dancing and there are different loops that are coming in contact with the DNA. And essentially, this helps to pull apart the single strands of DNA, causing translocation of DNA. All right. So that I will stop here, and we will continue a little bit to try and understand how the helicases can function as a motor. So when we say motor, we think about step sizes, we think about general terminology. So how is it done? How can you do a single molecule analysis of helicases and so on? So we will have the next class tomorrow. Friday, we do not have a class, and neither do we have it on Saturday. We have the next class on Tuesday. And Wednesday, this is an announcement. Wednesday, we'll have a special class where we will have an expert in the field of single molecule biophysics explaining to us about magnetic tweezers. So we have read about optical tweezers, we have understood what's optical tweezers. We will have an expert telling us about magnetic tweezers. So please do attend this. It will be really informational, and we will have Dr. Saurav Raj explaining about magnetic tweezers to us next week on Wednesday. All right, we will continue the class tomorrow at 8 a.m. Thank you. I'm here for 2-3 minutes if you have any questions.
Ma'am, I have a question. Sir, ma'am, in our project one, you mentioned that